In this video, we will going to learn about what are the different types of COVID-19 tests. Starting up with PCR. PCR stands for polymerase chain reaction. So in PCR, we will going to start up with obtaining the specimen, a swab, from an area which is called a nasopharynx. So the specimen has to obtain from this particular area. The second step is to extract RNA from the specimen and convert it to a DNA using a specific kind of enzyme which is called RNA polymerase. This enzyme will convert an RNA because, as we know, COVID-19 virus has the form RNA, so we have to convert it into DNA using this enzyme. The third step is to amplify the DNA of the virus and interpret the result, whether it's positive or negative. The second kind of test is called a lateral flow test, and it's kind of has like a similarities to the PCR. It has also the advantage, it's like a rapid kind of test, but comparing like the two, PCR is better than lateral flow tests because like we have more accuracy in PCR. In this picture right here, we have a machine called real-time PCR. And the real-time PCR actually makes our life easier nowadays, because in the previous years, scientists used like tedious ways in order to perform this kind of test. But this kind of like machine makes our life very easy and smooth, and you can get result. Maybe it depends, but during like half an hour your result going to be ready. Moving on to an antibody test. An antibody, actually the professional will like detect the presence of the antibody, whether you have the antibody, and having the antibody means that you have the immunity. So we have to differentiate between antibody test and PCR. What is the main difference right here? Antibody test will, as we said, it will detect the antibody. That means this test can't detect whether you have the virus or not. And that's because it does not detect like the presence of DNA. So that's an overall like view of these kind of tests. I hope that you enjoy this video.